Doing good. So, understanding pyrosequencing. Pyrosequencing is a method of DNA sequencing or determining the order of nucleotides in DNA, which is basically DNA sequencing is. Based on the this is based on the sequencing by synthesis principle, in which the sequencing is performed by detecting the nucleotide incorporated by a DNA polymerase. Pyrosequencing relies on the light detection based on the chain reaction when pyrophosphate is released. A Swedish company known as Pyrosequencing marked this method for short fragments. So basically, pyros in Greek it means as fire because light is produced in this process. Further, pyrosequencing biochemistry. So here we are going to learn about the biochemistry behind this process. In DNA synthesis, a DNPT is attached to the 3' end of the growing DNA strand. The two phosphates on the end are released as pyrophosphates, PPI. Sulfurase uses PPI and adenosine 5' phosphosulfate, which is known as APS, to make ATP. We can also see over here in this diagram, like it utilizes PPI and forms ATP in the presence of ATP sulfurase. Then luciferase is the enzyme that causes fireflies to glow. It uses luciferin and ATP as substrates, converting luciferin to oxyluciferin and releases visible light. So the amount of light released is directly proportional to the number of nucleotides added to the new DNA strand. After the reaction is completed, a pyrase that is used to that is added to destroy any leftover DNPT. Now let us learn something about luciferase enzyme. Luciferase is the generic term, sorry, for the class of oxidative enzymes that produce bioluminescence and is usually distinguished from a photoprotein. Several organisms such as American firefly and poisonous jack-o'-lantern mushroom produces this enzyme. Uh, as we see in their images, that they produce this type of lights. General working scheme for this process: DNA fragments is amplified with PCR. One of the two primers used is labeled with biotin. For streptavidin bound beads are taken. Amplified DNA is coupled with the beads and denatured with NaOH. NaOH increases the pH of the solution leads to the breaking of the hydrogen bond. That's why we are denaturing with NaOH. Then the biotin labeled chain remains bound to petals. Now the washing steps carries on. Complementary chain is washed away and a single strand left that is connected with the biotin or biotin label. Template is pipetted onto a microtator plate that contains sequencing primer, substrate mixture. In substrate mixture, there is DNA polymerase, ATP sulfurase, luciferase, and apirase are added. Now, moving ahead, the preparation is placed in the machine. The primer is first hybridized on the template, and this is followed by a gradual DNA synthesis. As a new strand is being made, the order in which the deoxynucleotides are incorporated is detected. So the sequence can be read as the reaction proceeds. The machine adds one of the four nucleotide triphosphates and lets the reaction take its course. If the DNPT address is complementary to the next base of the template, it is inserted by the DNA polymerase and a pyrophosphate, that is PPI, molecule is released. The addition of the deoxynucleotide to the end of the growing strand is detectable because it is accompanied by a release of molecule of pyrophosphate, which convert the enzyme sulfurylase into a ATP sulfurylase into a flash of chemiluminescence. Now these are the reactions involved in this process. Like when we add a nucleotide, it releases PPI, there's pyrophosphate. Then that pyrophosphate with APS, adenosine pyrosulfate with ATP sulfurase enzyme gives ATP, and ATP with luciferin in the presence of luciferase enzyme converts into luciferin. and produces light, from which we can detect the we can detect. And these are the reaction in which a pyrase enzyme degrades the remain DNPTs. Now, moving ahead on the general working scheme, the light prediction is recorded during the entire procedure by means of the camera, means of a camera. It is quantified, evaluated, and presented in the form of signal peaks, which we can see in this picture over here. If two or three of the same nucleotides follow one another in the template, several nucleotides will be installed in a single round, and the light production will be twofold or threefold as high as normal. Like here we can see, here we used GG or CC, so the we can see the peak is threefold and twofold. Like it depends upon how many nucleotides we are using. Now, if no nucleotide is inserted, it will remain pitch black in the well, and there is no signal peak on the graph, as we can see it in the case of here. See over here that we have used no nucleotide, and we here no peak is seen. Signal peak is no signal peak is seen. Now, of course, if all the four deoxynucleotides were added at once, then flash of light would be seen all the time and no useful sequence information would be obtained. So each denucleotide is therefore added separately, one after the other, with a nucleodase enzyme also present in the reaction mixture so that if a deoxynucleotide is not incorporated into the polynucleotide, then it is rapidly degraded before the next one is added, so that we can detect easily that which nucleotide is added and of which the light is indicating. Now here we are going through some methods of pyrosequencing. So the first method is and the most ancient is solid phase. The principle of pyrosequencing was first. This principle of pyrosequencing was first discovered in 1993, but combining the solid phase sequencing method with stephetavudin coated magnetic beads. And in this process, in this phase, the DNA is immobilized, and we are using three enzymes in this process and wash step to remove nucleotides after each addition. So washing step is important in this process. So here the question arises: why streptavidin? Sorry, because streptavidin has an extraordinarily high affinity for biotin, which is also known as vitamin H, and binding of biotin to streptavidin is one of the strongest non-covalent interaction known in nature. So moving ahead, there is a second method, which is liquid phase. A second solution-based method for pyrosequencing was described in 1998, and is the additional and in this additional enzyme, a pyrase is introduced. In this, there are a use of four enzymes, and the pyrase is the new one, that is nucleotide degradation enzyme. So this whole process is carried out in the liquid phase. That's why we are adding a pyrase over here. Otherwise, washing process carried out the whole process in the solid phase. Like in this elimination need for washing step. So there is no need for washing step in this method. Now, the third method is 454 technology. This is the newest one. The third microfluid variant of the pyrosequencing method was described in 2005 at the company 454 Life Sciences. This approach was based on the original principle of attaching the DNA to the sequence to a solid support, and they showed that sequencing could be performed in a highly parallel manner using a microfabricated microarray. So here are some cause and pros of the pyrosequencing. Some advantages are like a rapid method for the analyze of individual base polymorphism with a high throughput up to 4,500 tests can be analyzed, and with a second related system, you can sequence up to 48,000 probes per day. This process is accurate. Parallel processing can be done, or this can be easily automated. No need for a gel electrophoresis. There is no need for electrophoresis. These are some disadvantages. There is not a procedure for a simple research laboratory. Like Simple research laboratories cannot go through this process. And this work required a considerable and a special software is required for the instrument and for the analysis. And the third one is the complete system for the following process is so expensive. Like pyrosequencing is going to be so expensive sometimes. Uh, that's it. Thank you.